Your brain is a miracle. It stores your memories, creates your feelings, and forms your thoughts. 100 billion nerve cells control everything that happens in your body. But your brain is also susceptible to diseases that can kill these nerve cells. Neurodegenerative diseases like dementia. And when your brain dies, you die too. We live in a time when tremendous progress is ongoing. Research has helped us conquer many diseases, and our modern way of life enables us to live longer. But none of this matters when it comes to dementia, where our bodies are sustained, but our brains are not. Today, more elderly people die of dementia than cancer. So what can we do to stay healthy longer? What can science come up with so that we can be 100 years old and still be healthy in both body and mind? Today in Sweden, researchers are hard at work to help your brain's future. We start in Umeå. Memory plays an important part in so much of our lives. Everything from childhood memories, to how to ride a bike, to where you left your keys. Lars Nyberg is a scientist interested in how memory works and how it ages. Because when memory changes, it changes who we are.
If we lose our memories, to some degree I guess one can say that we also lose ourselves. We will lose our ability to remember what happened yesterday, the day before, and to some degree also our ability to plan for the future.
What affects memory? And is there maybe something you can do to protect your memory?
Hej! Är det Madeleine?
Ja.
Hej Mikael, välkommen.
Tack.
Då ska du få hänga av det här.
At the University of Umeå, there is a unique memory study, the Pernilla Project, which has been ongoing for 30 years, following memory development in many people over a long period of time.
Ja, så att idag då så kommer du få göra ett test och det kommer gå till så att du ska få lära dig ansikten och namn.
Nearly 5,000 subjects are included in the study, returning every 5 years to perform tests on their memory.
Så vill jag att du trycker en gång med pekfingret, bara för att visa att du har sett ansiktet.
For Madeleine, this is the first time.
Då tar vi och kör igång. Är du redo med handen där? Ja.
The main focus of the project is in what is called episodic memory. As the name suggests, it's about memory of episodes or events that have been experienced. It could be what you ate for breakfast, who you met at the store on Friday, or what you received for presents last Christmas. Previously, it was thought that this memory had already begun to deteriorate in people in their 30s.
The findings from the Pernilla project, they deviate from international findings in that we find that on average, episodic decline is seen not until age 60. We also see that many older adults have a very well-preserved memory. We find that some 80-year-olds have as good a memory as 30, 40-year-olds.
In addition to memory exercises, participants also go through brain imaging. Here, the research team wants to see how the brain cells work when exposed to different tasks.
Så där Madeleine, då ska du få ta och sätta igång det här testet med siffror då. Så att då ska du bara svara ja eller nej med pekfingret eller långfingret.
Ja.
Using functional magnetic resonance imaging, we can see that certain individuals who have problems with their episodic memory, they recruit certain brain regions of importance for episodic memory to a lesser degree than those who have an intact memory.
For many of the Pernilla Project's participants, dementia has developed during the test period. Why is it that some of the elderly are showing signs of changes in memory, while others have the same quality of memory they did when they were young?
certain lifestyle factors are important. Sometimes we use the term use it or lose it to say that one should continue to challenge the brain also in older age, but also activate yourself in other ways. Continue walk, continue bike, whatever you can do, and also eat well. Certain nutritions seem also to go together with well-preserved memory in aging.
How we live our lives affects how our brains age. But that's not to say you can control everything yourself. Your genetic inheritance is also an important puzzle piece for the condition of your brain. Just a few floors up from the MRI, the pursuit of the genes responsible is underway.
Hvor er Lotta? Pernilla skal gå og søge efter folk.
Here, the intensive search is ongoing.
Er hun nede på plan 2? Nå, der er Lotta. Bra!
Not just for colleagues, but primarily tracking down the genes responsible for the disease.
Og du skal vise.
Nu ska jag visa en ny mutation som jag hittar här. Där, ser du den?
Peter Andersen and his research team here at Umeå University are on the path to find the cause of the brain disease ALS. They are looking for the genes that create the disease, but in addition, they are also looking for the opposite, those genes that protect against the disease.
We want to find those neuroprotective genes because they are an obvious target for the development of new drugs for treating ALS.
Peter doesn't only work as a researcher, he's also a doctor and meets with his patients every day.
Hej, välkommen.
Tack.
Hur är läget?
Ja, det är inte så farligt.
One patient he's known the longest is Anna-Karin Karlsson. She has a hereditary variety of ALS, a disease that affects the nerves that control the muscles of the body.
Okej, ska jag pröva med din koll? Ja. Du har svaga reflexer idag. Det är klassiskt.
Värsta är ju...
Ja.
Men själva muskelsvagheten börjar komma 94.
Ja.
Men jag följer omkull och jag ramlar ju tidigare.
Ja, dålig balans.
Ja.
Ja.
Peter's team quickly found the gene that caused Anna-Karin's ALS. But something was strange about the progression of Anna-Karin's disease.
Där måste man ju säga att du har klarat det långt bättre än genomsnittspatienten.
ALS is a fatal disease. Life expectancy is only 2 to 3 years after the disease starts. But Anna-Karin is living much longer.
Ja, det tror vi ju beror på att du förutom att du har ett sjukdomsanlag så har du också motverkar sjukdomen.
Can such guardian genes exist? Yes, and Anna-Karin is not the first case.
This is a pedigree of the world's largest ALS FTD family. It's a Swedish family from the county of Västerbotten in northern Sweden.
The researchers in Umeå have managed to chart the ALS that runs in this specific family all the way back to 1886. But here again, it was strange that most of the family did not suffer from the disease. Only the ones marked in color became ill. Something protected the others.
So this shows that in addition to a disease-causing gene, there must also be very strong modifying gene which somehow protect individuals who are gene carriers to become affected. They are, let's say, nature's way of showing us the fastest way to new medications.
In his lab, Peter's research team is working in the search for the various ALS genes.
Hej Eva. Hej, kommer jag med lite prover?
The search starts with blood samples from patients and their relatives. And scientists have succeeded in localizing several different ALS genes.
25 years ago, we thought there might be 5 ALS genes, something like that. Today we know about 37. We know that there are definitely many more, because we have patients with known familial predisposition for ALS who do not have any mutations in any of the known identified ALS genes.
The list of ALS genes is growing, and at the same time, they have also been able to identify 5 protective genes. Anna Karin's protective gene, however, is still unknown. But Peter's team now wants to map her entire genome to solve the mystery.
Det känns bra. Jag hoppas de hittar något som hjälper andra. Kanske inte mig, men andra som kommer efter mig. Så det känns jättebra.
Protective genes are one way to bring forth new treatments against ALS. But if you should fall prey to the disease in the future, there is the problem that so many different genes can be found as the cause that everyone would need their own unique treatment. The dream would be to find a common denominator, and scientists are on this track.
As you can see, in all these neurons, you see these inclusions of misfolded SOD1.
It turns out that many different disease genes seem to give rise to the same defects in the cells, a protein that folds the wrong way.
If we can prove that there are common denominators for many types of ALS, perhaps all types of ALS, that is a huge advantage both for the patient community and for the development of new drugs.
Faulty folded proteins can be the key to ALS. To learn more about these proteins, we go now to Gothenburg. What is actually happening in the cells that leads to such devastating consequences? Not just with ALS, but in several of the neurological diseases that can affect your brain. Here at Chalmers University of Technology, we find researcher Pernilla Wittung Stafshede. She has been researching faulty proteins for a long time.
So I often use this toy to illustrate protein folding. So proteins normally fold to a compact structure that's functional, but sometimes they misfold, and that can lead to bad things.
It may look simple, but how this folding of proteins happens and what affects it can shed a crucial light on many of the diseases that affect the brain.
So this is interesting for many diseases: Parkinson's disease, Alzheimer's, Huntington, ALS, and also diabetes that come with age, because in all cases, protein misfold and stick together. So if we understand the basics of this, we can maybe cure all the diseases.
Proteins are contained within all the cells of the body. They are the workhorses and perform all of the important tasks in the cells. But they must have the right shape. When they are formed, they fold in a specific way. But like Pernilla says, sometimes the proteins fold wrong. Then they can no longer do their work. And what's worse, there is a risk that they stick with other proteins to form large clumps that end up killing the cell.
I want to understand how proteins fold and misfold and the basics of that, so we can learn eventually how to stop the diseases that are based on misfolding.
Pernilla's team is now growing bacteria that produce the protein they believe is behind the brain disease Parkinson's. In the lab, they will then try different ways of affecting the proteins.
We are mixing things with our protein to understand how those things affect misfolding, if they can speed up or if they can stop misfolding.
Pernilla's team now uses spectroscopy to see how their substance causes the proteins to fold. The result is shown as curves.
So this method probes the structure of the protein or the shape of the protein. And in the green sample here, we see it's an unfolded protein without structure. And in the blue one, you have a folded protein. But if the protein misfolds, that can result in that they stick together with other misfolded proteins. And then I need more. So then they stick together with others, and they can form long fibers, and that's toxic.
Here we have our last alpha-synuclein aggregation assay.
Yeah.
I already put the sample here.
To find out how these long fibers with clustered proteins look in detail, Pernilla's team now loads a so-called atomic force microscope with one of the samples.
These looks really cool.
Yes, and we got the 8 nanometer height, which is typical size.
So here we can see the amyloid fibers of our protein, and here we have some free fibers, and here we have a cluster of amyloid fibers. So this is very typical.
The microscopic images of the clustered proteins reveal how Pernilla's team has managed to influence the folding by adding their substance. And the researchers are coming closer to understanding how the wrong folding is occurring. This means that a cure could be within reach.
I'm sure we will find a cure for these diseases in the future. Many researchers study these processes in the world, and we all providing pieces to the puzzle. And it's not clear who will make the big discovery, but one of us will.
A drug may be on its way, but there too lies a big problem. If you become one of those who suffer from a dementia disease, billions of your brain cells will already be dead before you notice anything. The seemingly impossible task, then, is to treat the disease before it is even discovered. Here in a hockey hall outside Gothenburg, there is actually brain research going on. Researcher Henrik Zetterberg is working together with Swedish elite hockey players to reduce brain injuries caused in sports. Because diseases aren't the only things that damage brain cells. Concussions do too.
If you get a hard hit to the head, this hit will impact the brain tissue so that it might rotate or hit the skull. And then nerve cells might be disrupted and eventually they die.
Ja, det var en kille i mitt lag som fick en hjärnskakning för inte så länge sedan.
Ja, det hörde jag om faktiskt.
Så det var inget kul.
Nej, det var att han fick åka till sjukhus.
Ja, då åkte han, kom in ambulans in på isen.
Ja.
After a brain injury, the brain needs time to recover. That's why it's so important to be able to measure how much of the brain's cells are damaged. This isn't only important in the case of concussions.
My research is focused on the development of very sensitive and specific tests to detect brain injury in blood samples or cerebrospinal fluid samples. This is relevant not only to concussion but also to the many diseases that can affect the brain, like Alzheimer's, for example.
Välkommen. Ska jag ta lite benprover på dig? Så att om du kavlar upp armen där.
Here at Sahlgrenska University Hospital in Mölndal, Henrik's team is studying concussions along with our most common form of dementia, Alzheimer's.
Det sticker det.
Henrik's research team is developing a whole new method of measuring the degree of brain injury, using a simple blood sample.
The brain changes in Alzheimer's disease, they seem to appear many years before the first symptoms become evident. And some data actually indicate that it might be 20 to 30 years before the first symptoms appear. And then we have to develop techniques to be able to detect the brain changes before they injure too many neurons.
Blood samples are taken into the lab.
Och det är ett 50-tal prover här.
Ja, ungefär. Ja, 36 nästan.
36 är det, precis.
But in order to measure the number of damaged brain cells, Henrik needs to find a substance in the body that denotes brain damage, something called a biomarker. Henrik's team is now focusing on a protein found in brain cells. It acts as the cell's skeleton and is called a neurofilament. And when the brain cells are damaged, neurofilament leaks out into the brain and spreads into the spinal fluid and also into the blood. The level of neurofilament in the blood can describe the level of damage to the brain.
Nu håller jag på och laddar in informationen om plattan här. Vilka prover som är i vilka brunnar.
Just det.
But then it is up to Henrik to be able to reveal the proteins in the blood. And thus far, that has been impossible.
Men nu kommer du stoppa in proverna i instrumentet.
Nu är jag nöjd, så nu kan jag bara sätta in plattan här.
Now, Henrik's team is trying a new method that uses fluorescent antibodies that attach to every single protein.
This method is cool because it's so super sensitive. So we have struggled for many years to measure these brain damage biomarkers in blood, but we have failed. With this method, we can measure at ultra-sensitive concentrations, almost single molecules. And one could look at this as if you took a sugar cube, put it into 2 Olympic swimming pools and let it dissolve. And then you take 0.5 milliliter of sample of this water and measure the concentration of the sugar. This is as sensitive as this method is, and then we can measure the markers.
Här sitter du med resultaten, ja.
When researchers can see the damage to the brain's cells long before any symptoms occur, it will open the doors to giving new medicines in time.
Där är en som sticker ut, ja, just det. Ganska tydligt.
I am hopeful. I believe we will be able to develop effective therapies against Alzheimer's disease and other dementia-causing disorders. The important thing to be able to do this is that we can detect the diseases before too many neurons have been lost. And this is where these tests come into the picture. We have to be able to measure objectively that the brain changes are present and then treat those.
Pratar du om hjärnskakning i hockey någonting?
Ja, ibland om typ så här hur man kan förebygga och så.
Ja, men det har blivit bättre i elitserien faktiskt. För att man ser hårdare på tackling mot huvudet och att man då får matchstraff efteråt och sådant. Och då blir det färre farliga hjärnskakningar. Det är bra.
Henrik's research has gotten the hockey community to pay attention to the dangers of brain injury, and as a result, the number of brain injuries has already decreased. Research in the various dementia diseases is taking giant steps right now, so that your and everyone's brains can be healthy in the future. It concerns your own lifestyle, but also the secrets hidden in your genetic legacy, and how those genes affect the folding of proteins that can kill your brain cells. It's about detecting diseases in time, before causing serious damage to your brain. And if all goes as scientists are thinking, we will have the cure one day that can save your brain from the diseases that for now are incurable. And Anna-Karin Carlson, who has lived longer than most with her brain disease ALS, is following this research carefully, hoping for a breakthrough.
Det har hänt jättemycket. Det är ingenting som hjälper mig just nu. Men jag ser ju att de kommer framåt. Och de hittar saker som de inte hade en aning om för när jag blev sjuk. De måste få forska vidare. För det är ingen människa som vill gå igenom det här. Det är bara så.